Good afternoon. The truth is, to an audience of robotics researchers, our speaker today needs no introduction, but I'm going to give a brief one nonetheless. Russ Taylor did a bachelor's degree at Johns Hopkins, did a Ph.D. at Stanford, and then joined IBM Research, where he did a long list of things, but one of them was to lead the computer-assisted surgery group, which was responsible, among other things, for Robotic. He then went back to Johns Hopkins in 1995, where he's been the director of the Engineering Research Center for Computer-Integrated Surgical Systems and Technology, and is now going to speak to us about the sorts of things that he's done there. He is a fellow of the IEEE, among a number of other societies, recipient of the IEEE Robotics Pioneer Award, among a large number of other awards. So, Russ?
Okay. Well, thank you very, very much, Cam. I was just mentioning, I've been coming to CMU for various purposes for 30 years. And I think one of the first things that made me think seriously about moving to academia was I came to do a paper with Matt and Ken Goldberg, and realized that when I was coming I was happy and this was fun. Now IBM was fun too, but what I'd like to do first is to give, these are the usual obligatory thank yous and acknowledgments and disclaimers. And I'm going to be talking about work, a lot of it I've been personally involved with, but also work of my colleagues, and I've tried to indicate that on the slides. But this picture here, I was working at IBM on systems like this. In fact, this is a system I developed with Ralph Hollis. These were Ralph's fine positioning devices, and it had sub-micron precision over an area about the size of that tabletop, and we used it in manufacturing processes, and this is, of course, Robotic. Now, these systems, they look really pretty different, but I think the underlying insight that made me begin to be interested in systems like this is they have something in common. What they do is they couple information to action, often in partnership with humans, to fundamentally change a process. And it is really my belief that this interventional medical computing can have the same kind of impact on medicine that it's had on manufacturing. And perhaps by the end of the talk, if you don't already believe it, you might at least agree that it's plausible. This picture here is very similar to a picture that I first put together for my bosses at IBM, trying to explain why a computing company should care about something like medical robots. And if you look, the notion is you start with everything you know about the patient and everything you know about people in general in surgical procedures. You mush this together into some kind of a computational representation, and I've called that a model. It may or may not be an accurate geometric description, but a representation of this information that lets you do what you need to do. And the first thing is you need to diagnose the patient's condition and plan your intervention. Well, once you've made the perfect plan, what you really have to do is execute it. So the idea is all of this information comes into the operating room or interventional suite, where once you've kind of fused the model to the physical reality, you can then use appropriate technology to help you do what you plan to do and to verify that you've done it. Well, we're engineers, so what is that? That's a control loop tied to a particular patient's intervention. And you notice this picture here is replicated, and that really is to remind us that this basic process, information process, actually occurs at many, many, many different time scales, from weeks and months for a patient management down to second by second in the operating room. Now, there was something else in manufacturing that we did. You have here an information-controlled process that helps you be consistent. And because it's information-controlled, you actually maybe know what you did. There's no law written in heaven you have to throw the data on the floor. So the other thing that we did in manufacturing that was as much a motivation to me, even from the beginning, was this. To take this information, eventually you know what happened with the patient. If you know what you did, perhaps you can relate the two and more effectively improve your treatment processes. So this is basically surgical CAD/CAM and surgical total quality management. And my basic hypothesis is that it is really the synergy, it is these two loops and their synergy that has the potential to make a profound difference in the practice of medicine. Well, actually, that's a pretty old idea. This is Imhotep, who was generally believed to be the father of surgery. And Howie, you didn't meet him in Egypt, I guess. But, what he taught was a highly empirical process. There weren't computers, but it involved basically all of those steps. But of course, the difference is the power of computing and equipment interface to computing can make that process much more effective. So just a couple examples, and I'll come back and talk about them. A lot of the work that we've done over the years has involved placing needles in a minimally invasive fashion very accurately on anatomical targets that you can see in medical images. Here's another set of examples in which typically you're doing very highly interactive surgery, such as microsurgery, or these are the Da Vinci robot. And what we're doing is we're using this information very interactively with the surgeon in the operating room. And you can think of this as surgical assistance. But they're the same basic building blocks put together slightly differently for different applications. Which leads me to my next point, if I can make this slide. Come on, guy. Bill the Gates, what are you doing to me? Okay, thank you. Whoa. I have no idea what's happening. Sometimes if I care about running a thing without, okay. So here the idea is to build this. We're fundamentally talking about systems. And there's a lot of parts. There are a bunch of things. These are buzzwords that we have in medical image processing and modeling. The computing is ubiquitous. We're also dealing with the physical world, so the interfaces between the data world and the surgeon are important, and there's a science to systems. Medical systems especially, there's some very important systems constraints. So you need to think about all of these things together. And that was the motivation that really led us to propose and get funded this engineering research center for computer integrated surgery, where Carnegie Mellon was one of the charter core members of this partnership over the years. And generally, I'm going to be using examples from a very broad set of research, all focused on systems in the balance of my talk. We spent what was maybe by Robotics Institute 1 year's budget, but we spent a lot of money over about 10 years of seed money, and we're now self-sustaining. Just to show, these are the pictures from our first site visit, where the goal of these engineering research centers is you should be making contributions, sort of basic science and technical artifacts and systems, and using these different levels to motivate and look at the synergies between these levels. And this is the last time I bothered to make this picture. And we've done a lot, and I can't talk about it all. So what I would like to do for the balance of my talk is to, oh, where did my slide vanish, is to go back and use this picture as the organizing principle for what I want to talk about. And the first piece of that is patient-specific modeling. A lot of this work is based on medical imaging, and there's a very, very broad research agenda here and that we've worked on over the years. I want to just, as an example, pull out one common thread that has been quite important for our research and the communities over especially about the last 5-6 years. And that is where you have previous information about a patient, either like CAT scans of the patient, or you usually know if it's a human you're operating on, and you know something about how people's anatomy varies. And now you get some new information in the form of images. So what you want to do is mush all of this information together to make a better representation of the patient for whatever it is you have to do right now. And often you're doing that actually during the intervention itself, in other cases in planning the intervention. Just a few examples. Here is one. This is some work I had some involvement with, but what we have here is a patient with a kidney tumor, and we want to cut the tumor out while leaving the rest of the kidney as much as possible undamaged. And so the surgeon needs to know where to cut. So what we took is stereo video from this Da Vinci surgical robot, made a 3D representation of the surface of the kidney, fused that information, registered it with a preoperative CT model where we knew where the tumor was, and then generate various graphical overlays to tell the surgeon, here is where things that you can't see directly because they're below the surface are, like the tumor. The red is the cut here line, the yellow are the filling system of the kidney. Another example, we've done quite a lot of work motivated mainly by orthopedic applications, where what we're doing is we know we've got a human, but we only have a few X-ray images, but we want to get a good 3D model of the person, or a best guess as to what the CAT scan would have been if we could take a CAT scan right now. And this has become a huge cottage industry in medical imaging. We did some of the earlier work, and there's a large community. Here's one example of how this works. Basically, here is just sort of an edge map of what is really a 3D volumetric density model, and you get rough alignment. Now we're adjusting each of the femurs to be their most likely, and then we're kind of re-registering it all together. And you can get models that are good to about 2 millimeters from a small number of images. And this is typically good enough for the kind of thing you need to do in planning or navigating for orthopedic surgery. One use for planning is there's an orthopedic procedure called an osteotomy, and this is a hip osteotomy where there's a cartilage defect or other defect in the hip, and what you're doing is you cut the socket part of the hip joint free of the rest of the pelvis. You move it and you bolt it together with $50 or $500 straps of metal and $700 sheet metal screws. And the point is that one of the common cases are with young women with hip dysplasia. Now with young women, you really don't want to use a lot of radiation in their pelvic region if you can help it. So what we're doing is we do take a CT just basically of the socket, because we want that accurate, and then combine it with just a couple X-ray images and statistical information, and by mushing this together we can get quite a good representation that is plenty accurate, both for planning and then for helping us execute this osteotomy. Another example In the operating room, if you take x-rays, 100 x-rays from all around the patient, you can basically do a CT-like reconstruction of the patient. The problem is, to do it accurately, you need to get all of the different projection directions, and that is not always practical in the operating room for a lot of reasons. So what we did, again, is we take a prior CT or statistical information, take whatever x-rays we have, make our best guess as to the model, and then use the model to fill in any missing x-rays. And we call this hybrid reconstruction. And again, it produces significantly better reconstructions that are good enough to provide effective intraoperative feedback to the surgeon. One other example here is this is actually a screening application. If you have osteoporosis, people want to do a bone scan, a dual energy x-ray, and then compute your fracture risk, which is basically a biomechanical beam. Basically, it's the strength of a funny shaped beam calculation you're doing. It's fundamentally a 3D calculation. And, but you can't really do a CT scan 2 times a year for purpose of screening. So what we do is we take a small number, in this case 4 of these DEXA images, combine it with a statistical model, and make our best guess as to what the CT would have been. And then if you do biomechanical analysis on our best guess CT, and compare that to the biomechanical analysis you would do on a real CT, you get correlations better than 0.9. So that's very encouraging. So these are just, I'm trying to give just a quick overview, and I don't know how long I will wind up speaking, but that is some of the flavor of the imaging and modeling component of what we're doing. Sometimes it's preoperative imaging, sometimes it's intraoperative imaging, and there are a few other examples that you'll see kind of sprinkled through the talk. Planning, I'm not going to say a lot about. It's clearly pretty crucial. Sometimes you're doing it on the fly. An awful lot depends on the particular application. Like here, you're doing biomechanical analysis. This is that osteotomy. Here, what we have is statistics of where cancer is likely to be found in the prostate. And if you want to do a biopsy to sample the prostate to see is there cancer there, you want to pick the biopsy sites that minimize the chance of a false negative. And so you have to take these statistics and register it with a real prostate. Here's an example, safety barriers in a neurosurgery application I have a slide about later. Execution, well, this is the Robotics Institute, and robots have the best movies. So well, let me talk a little bit about some of the robotics-oriented work. Again, there's a lot. What we're fundamentally dealing with is coupling information to action in the physical world. And you can think of the robot as the one direction, but also sensors coming back the other direction. It's that whole interface between the data world and the world that we're dealing with here. Okay. Oh, this slide is out of order. Well, let me talk about it. What we have here is we want to biopsy. Again, this is a big kidney tumor with the robot in a CT scanner. This is actually one of the early applications. This is maybe, well, 8 or 9 years ago. And what you want to do, this is CT fluoroscopy. You can see the target in the image. But what you want to do is use the image actually to tell you where the robot is too. Now, in this case, the fluoroscopy, you can see the needle going in, but oftentimes the needle isn't actually lined up with the target. And so you need some way either to line up the needle path so you can see it in the image, or at least know where the needle is. And so to do that, what we do is we put a fiducial structure on our needle driver so that from the pattern of even from a single image, we know exactly where that needle is, and then we can do even out-of-plane biopsies or visual servoing to hit a target. But this is really to remind us, and this does come out too dark, I apologize. We don't always need a robot. And George Stetton has pursued a similar idea here. Actually, I think the first uses were actually at Hopkins and at Tokyo. And what we've got is an image overlay device where if you have a display and a mirror, you can line things up so the scan plane, the virtual image of what you're seeing in the display corresponds to the physical location in the patient. Then you just use your hand-eye coordination to do a biopsy. And so it's hard to see. This is an MRI version. This is a CT version of that idea. And this is just finally in clinical, going into a clinical trial. Here's another example of placing a needle on a target under image guidance. This is a very simple robot basically for prostate brachytherapy. The standard way that you do brachytherapy is you plan a pattern of radioactive seeds you're going to put into the prostate, and then they have needles that you insert into a grid that looks like this, one at a time with the seeds in them, and then you withdraw the needle, leaving the seeds behind. The problem with that is you're restricted to the grid. It's kind of tedious because you have to line things up. And further, the paths are all constrained to go straight. Well, that's a problem because with a patient with a large prostate, you often can't hit where you want to go because the pubic bone is in the way. So here, this is a very simple robot that positions a needle guide and is all registered to the ultrasound, which is giving us the image feedback in an integrated system. And we've done, I believe, 2 clinical trials on this right now. In fact, there was just a clinical experiment yesterday or Wednesday on this.
The engineer design. So I was trying to figure out, the needle goes into the person, or?
Yeah, the needle goes into the perineum. This is the space between basically the anus and the base of the penis. So it goes in.
What's the white tube for?
This?
Yeah, this is.
This is the ultrasound probe.
Oh, I see. So
this is the needle guide. So this is in the patient's rectum.
Right. And which, I don't know how to ask this question politely, so I'll just do my best.
Yeah.
Which way is up?
That way. Here's the robot. I'm not sure if this is a mannequin or an actual patient. They've done a lot of actual patients, but it's a fairly gross picture. We don't normally. Now, there is a lot of interest in doing both biopsy and brachytherapy in MRI environment. Now, in MRI, of course, materials become quite an issue, and so here is a joint project we have with the Brigham to develop, I need newer pictures, a robot that's completely air actuated for, again, brachytherapy and biopsies in MRI. The thing that doesn't really show here is the actual integration of the control. So again, there's sort of direct feedback from the MRI scanner. Here's another robot. This is more for biopsy or just injections into the prostate. Again, under MRI guidance. So here we have an imaging coil and a needle guide that is kind of steerable. And then originally it's just sort of a manually actuated remote mechanism for aligning the needle to hit the target. The really cool thing to me about this is in less than 2 years it went from literally the back of a placemat sketch to a clinical trial. Further, the system has been commercialized. Well, here, I'll show the commercial in a moment. Here, this is some of the earlier states. Here, this is a prostate. Here are targets. And then here we're imaging after where the targets were after the void is where the needle went. And you can see it's really very accurate. The device, a really simple device, has been commercialized by this company. And this is their first prototype. It's the same idea. It's sleeker, better. You're getting good targeting accuracy, and they've got an amazing imaging coil. Having an imaging coil right close to your target gives you much better imaging in MRI. And now there's ongoing research. Louis Whitcomb and Julian Urartikita are developing now an actuated version of this same thing. Those are all examples, and I've got 40 more I could give you. We talk about needle placement, but they're really needle aiming robots. If you have a skinny needle, one of the problems is once you start poking it into something, it's going to bend, it's going to deflect. And one of the reasons for that is most of these needles have bevels in them, or little hooks, and you can exploit that. As you advance the needle, if you twist it, you can steer it like a bicycle. And so Alison Nakamura has led the Hopkins effort in this area. Cam is also, with a little bit different design, has been pursuing similar work here. And I think yours is neurosurgery, I think, isn't it, Cam? You still doing it? Yes. So if you're interested, it's a cool thing. And if you're interested, you've got someone local, or you can come talk to folks here. One of the challenges with things like this Da Vinci robot that everyone has seen is you have marvelous dexterity, but once you get smaller than 5 millimeters, just the mechanisms become harder. You know, there are all these little bearings, pulleys, pivots. The mechanism gets really hard, especially if you want to keep it strong. So one thing that this is work that Nabil Simon and I did is you'd like to replace all of that mechanism with just simple bending metal. So what we have here, if you take two pieces of metal and you kind of tie them parallel, I push on the one at the top and pull on the one at the bottom, I'll get bending. Further, those two pieces of metal can be my structural elements. So here what we have is we have 3 pieces of metal. You then get bending in 2 directions, so we have 3 tubes. It's a little girder, and running through each tube is wire. So we have 2 stages, each with 2 degrees of bending, and then down the middle we just put an actuator for a little gripper. Actually, what we found out, much to our dismay, is that the gripper is very hard to design to make one that works. And that actually set us back by about 8 months as we went through gripper designs before we could begin to test things of this sort. This has now been licensed to a large medical robotics company who I will not name, but most people could probably guess. And here again, you can kind of see the sort of shapes you get. Here's another project that we're doing right now. And the goal is the motivating application is on the next slide, and let me do this in another order. What we would like to do is get into a confined space. This is an osteolytic lesion, and basically clean that space out from the inside. What has happened is the bone here is deteriorated. It will continue to deteriorate unless you can clean it all out and then fill it with cement. There's a similar problem that this is from wear particles in the cup liner of orthopedic implant. You can get similar problems in bone cancer. And so what we're after is a minimally invasive way of cleaning that out, but you still have to exert a lot of force and have a large family of tools. So we needed a large lumen with quite a stiff and strong mechanism, and again, something that we could mass fabricate without mechanism. And so what we have here, it's a very clever way of getting steel guide wires through embedded in a 1 millimeter wall thickness tube. And so we have an 8 millimeter device with a 6 millimeter lumen, and it's pretty strong. Can show. So this is just going to show some of the strength. So what this is showing is that the units don't match, that you get, you know, for roughly 7.5 pounds, it's maybe 5/10 or 6/10 millimeter. So it's quite stiff. Now, come on, guy. So, and now you can also build these things. This is just a new prototype where these are doing S-bends. And we're looking at a lot of applications beyond orthopedics, but basically places where you need quite high stiffness and strength in your distal mechanism with a large lumen.
So Russ,
Yeah.
I'm sorry to interrupt you. How does that work? How do you look at the shapes?
Pardon? Oh, the shapes are very simple. This is more, you're pulling against a spring, and they're guide wires that just run through the, there's one set of wires that end here, and then a parallel set that go to there.
Oh, so there are 2 stages?
Yeah, again, this is a 2-stage snake.
I see how.
And the key point about this is actually the fabrication process is somewhat scalable, and there are other design, and basically we can modify the design basically by changing the flexure pattern.
And does the diameter go down as you add subsequent stages?
Well, that one, no, that one is that's one tube with 2 sets of guide wires terminated at different places. There are a lot of options. I mean, that's just kind of a very first prototype where we're putting something together for this osteolysis application, but there are a lot of other applications. Here's another example. Again, this is sort of some system work that we did. Again, with this notion of human-machine partnership, dexterity enhancement. And here, of course, this is the Da Vinci surgical robot, this remotely actuated robot. And what we were interested in doing is giving the surgeon the ability to use laparoscopic ultrasound with some of the same dexterity as if it were open. The point about the Da Vinci is the surgeon sits, looks into stereo video, controls these control handles, and it kind of feels to the surgeon like he's manipulating his wrist inside the patient. So what we'd like him also to be able to do is manipulate ultrasound inside the patient. And so we developed this. They developed the tool, and we basically developed the software and the interfaces to the tool. And this is all built on an open source software environment that we're actually looking for more people to collaborate with. I'll say a bit more about that at the end. One of the things, this kind of is how we might want to fuse that Da Vinci project that video Da Vinci project with this ultrasound. There is a technique I'll say more about later called elastography, where if you push on tissue with ultrasound, with an ultrasound transducer, the speckle moves with the tissue. And by looking how much the speckle moves as you push, you can compute the relative stiffness of tissue. And that will often give you better contrast if you're trying to find anatomical structures than plain old B-mode ultrasound. You can also often see a tumor, because tumors are often different stiffnesses than, with elastography than that you couldn't see in standard B-mode ultrasound. So the idea is to use either freehand palpation or the Da Vinci robot to get these elastography images, register those to preoperative CT, then generate an intraoperative visualization. Here, one of the other things, as you're doing, once you start cutting into the tumor, it kind of all flops, the kidney, it all flops around, and you've lost all of that nice preoperative data. Maybe you could use like one of these new Kinects to kind of track how all of that tissue is going. But in this case, what you have is you want to keep the tumor as a coherent meatball with kind of a coating around it of about 5 millimeters of healthy tissue. So what we do is after we've done the registration, we put an electromagnetic marker into the tumor. Then the idea is once you start dissecting the tumor away, the tumor stays intact and you can still have a visualization as to where it is. So we've done kind of this level of lab experiment. We're currently putting the elastographic capability onto the Da Vinci, and then the goal is to do these things together. How do you feed back information to the surgeon for things like forces or stiffnesses or other information? You can try to feed them back directly haptically. It turns out it's really hard, especially if the master device wasn't designed. And often it's also not too popular with surgeons, especially for some of the microsurgery applications I'll talk about later. Surgeons don't really like you pushing on their hands. So here the idea is to use, in this case, graphic visualization as a sensory substitution. And this is these examples are very largely the work of Alison Nakamura and her student Tomo Yamamoto. And here, this is just force. Here, what she's doing, or what they're doing, is they're palpating to determine relative stiffness and then feeding it back again just by showing a surgeon a visualization of the relative stiffness of the tissue. This is personally my largest single project, and it's one that I feel really lucky to be collaborating with Cam on. And this is vitreoretinal microsurgery. In fact, we now have actually several existing or prior grants with Cam's lab on various aspects of this project. And this is the most difficult technically probably the most difficult surgery that anyone does. The surgeon this is a large instrument. It's 0.7 of a millimeter across. Some of them are 0.5 millimeter across or smaller. The surgeon stares through a microscope. This is what the surgeon sees. It's a little brighter. Some of this is just this projector, but you don't really see a lot. You're staring, you're holding your neck really rigid and getting neck and back pain. And so when we put all of this together, it's technically extremely difficult, and the surgeon really can't feel the forces that he's interacting with, and really it has a hard time knowing what's actually happening between the tool and the tissue. And by the way, his hand shakes. So we're trying to overcome a number of those limits. And so the flagship grant at Hopkins for this is one of these BRPs, where the goal is really to put together a complete system. This is an earlier version of that system, but the basic components are here. you take a surgical microscope and equip it with stereo video, so this, and a video display. So this is a stereo video microscope. What you don't see, which is the true heart of it, is really the computer. And then there are various manipulation aids and smart tools that kind of are all plug-ins for this basic system. A big part of what we're doing is computer vision. You need to track in stereo in real time these tools relative to the retina. It's a little hard to see, but also track the motion of the retina. Once you've done something here or made a measurement, as the retina moves around, you want to know how it's changed. This is just some real recent work from Rogerio Richa, who is a postdoc with us. But here he's tracking the tool and giving, when the tool gets within a millimeter of the retina, you don't want to warn the surgeon, but really, because the surgeon will get mad, but what you might really do is then change the control mode of the robot, if this is a robot, or turn a sensor on or off. Another problem in eye surgery is light toxicity. We've all gone to the eye doctor and had our eyes dilated, and then knows how it bothers us. If you shine too much light on the retina, you'll sunburn it, and you can actually cause a lot of even permanent damage. It turns out that the light that does that to you is more the blue light than red light or infrared light. And so the idea here is, if we're using video microscopy, we can manipulate the spectrum of the input light and then do various image processing tricks to still give us a full color visualization. And so in this example, this is some work of some of our students. Well, first what we do is we've built a RGB light source out of LEDs where these match the colors that you see with the video camera. And then you can modulate the light at a kilohertz. You can change color very, very quickly. And then one of the things you can do is, are these movies going to play? If you make every other frame say red light, and then you use the full color frames and then track the motion, you can then use where you've had full color information, you can fill in. And you can even do this with very significant reductions. Again, this projector is projecting colors a little off. And this movie here is about a year old. There's been some progress since. But this is the kind of thing you want to do. Now, one of the interesting challenges is you'd like to maybe have the computer automatically decide when the surgeon really needs high fidelity color and when he doesn't, and have it automatically, based on understanding the task, modulate. Now, hand trimmer. Surgeon's handshake. And Cam, have you given seminar to this group? Yeah, so this is the Micron device, which is where again, you can actively cancel the hand trimmer. So one of our goals is to incorporate this sort of freehand device into our total system, and then use various forms of in-point sensing, video processing, or sensing built into the tool to help us control the robot to reduce tremor. The other mechanism we have for this is what we call the steady hand robot. And here, it's just as easy for me to show here. The idea here is the surgeon and the robot both hold the tool. And the surgeon, the robot feels the surgeon pull on the tool and just follows his hand. And so there's absolutely zero hand tremor. Here, this is a 100 micron blood vessel that we're cannulating. This is a live video, but you can see the motion compared to freehand. And we're using both of these platforms for our research. Probably the micron will be quicker to get into clinical use because it's less of a disruption of the clinical workflow. Well, okay. Now, I mentioned that the surgeon can't feel the tools. I'm going to go a little over 4:30. Is that okay? Okay. If people walk out on me, that'll be fine. I'll talk to an empty hall. The forces are a factor of 10 smaller than you can really feel. I mean, Cam actually did some early work again here with his devices. So what we want to do is we want to build force sensors into the surgical tool. One of the problems is as the tool goes through the sclera, the force there is a lot higher than the force there. So the idea is to build the sensing down inside the eye. And to do that, what we've done is, these are large forces compared to what the thing can feel. We've been using these fiber Bragg grating, oh gee, it hid the sensor. The idea is this is an optical strain gauge, and as you pull on the sensor, you change the spacing of a grid, which changes the wavelength of reflected light. And these are excruciatingly precise. Our existing tool is about 400 micron, or I'm sorry, it's about the order of millinewtons. Now, the other problem, how do you feed that back? You can't, with a freehand tool, you can't pull on the tool, and the surgeon doesn't like it anyhow. So we've been looking at auditory sensory substitution. So you play a little tune that changes depending on how much force. And you can use speech. And graduate students can have fun at Johns Hopkins. Now, we've just started to use these sensors in some bunny experiments, but one of the other questions is, does that sensory substitution actually help you? And so here we've made a phantom so we could control the interaction forces and set what we thought was the tearaway force on the retina as a kind of a threshold, and then used auditory sensory substitution with a bunch of medical students and a few attendings. This is Dr. Handa, to see if the feedback actually helps. And this data is not yet published. It's not even completely analyzed, but preliminary data is pretty encouraging. Alarm, what that means is we ring the bell when we figure that you probably tore the retina. And if you give a warning before you're likely to do it, you can significantly reduce the number of times where you would have done damage, and you find that very quickly it doesn't take you any longer to do these peels, but it provides, but you can exert lower force while doing them. Actually, one of the things here that I think is most interesting is the potential, and then we're doing things like tissue force correction. One of the things I think is most interesting here is actually the potential of using that during training, to train someone what the right feel is, and even if you don't use it in routine clinical use. So they can view it as training wheels. One speculation is the new generation of surgeons may come to like the wheels. Another thing, if you can use force sensing in various ways to implement virtual fixtures and interactive behaviors, and for things like blunt dissection, you can make the robot just want to stay in a tissue plane. And here, this was something that we found really interesting. What we did is we had the robot be very compliant in a direction where it didn't feel like it had a lot of force. And then we're trying to tear a membrane. In this case, it's on an eggshell. And this is the path of the robot. If you look, it's hard to see, but just kind of naturally, the thing tore in a circular pattern. And now the eye surgeon got really excited when he saw that, because he said, look, that's just the motion we try to teach our residents to use for something called capsulorhexis, which is tearing sort of a membrane in the eye, as on the lens of the eye, as sort of the first step of cataract surgery. So this is kind of cool. Again, this is something that we're just starting to play with. Another kind of sensing is optical. You really don't know much about the structure of the retina right beyond the tool. There are a lot of reasons you might want to know that. So what we've done is we've been incorporating single optical fiber optical coherence tomography sensors into tools. This is a 0.5 millimeter surgical pick, and basically you can look beyond the tool to know where the tissue layers are. And then if you scan that across a piece of retina, you can see things. Like here is, again, I'm sorry, the projector is hiding all the detail, but this is a piece of a pig retina. And the really neat thing to us is we need to go back and do histology, but this, we think, is the internal limiting membrane of that eye, which is something you just really can't see other ways. The other thing is the particular form of OCT we're using. Also, you get the tissue color spectrum as sort of a side effect. And so you can then process the color spectrum to tell you, this is, again, you can't really see the detail, this is a blood vessel, and this is sort of the optical equivalent of B-mode ultrasound. And this is the blood in that vessel, and here what we've false colored it, say here's where we think oxygenated hemoglobin is. And again, this can be very useful functional information, yes?
Just to clarify, you're getting a single A-mode scan?
And then scanning it in this case, yeah. Yeah.
To get that image?
Yeah, and we're using common path Fourier domain OCT, if you know the buzzwords. So yeah, that's what we're doing here. This is largely the work of Jen Kang and his students. One of the things, the problem is you want to get this whole thing into a 0.5 millimeter surgical tool with room for the tool too. He also has imaging bundles that are a little bit larger, really just a bit too large for most optical retinal applications, but there what you have is a bundle, and you typically use a Galvo to scan the top end of the bundle. It's pretty cool though. And then, you know, you can take things and integrate them with the robot. Here is one example where we're saying, okay, maintain a constant distance from the target surface as it's moving. In this case, this is done with a rather slow robot. We've also looked at doing this with the Micron and a sort of a 1 degree of freedom version of the Micron that we put together at Hopkins specifically for this keep your distance application. Other things, you can do things with, if you have a robotic device, is you take a little scan, then you here's a target, you come back to the target, and then you want to make a very precisely controlled cut. And so this is from a couple years ago now, and so here is the profile of the tool, and here is the target. This is something that we're very anxious to do with the Micron as soon as we get the next generation of the Micron. I guess, Cam, we can try it with the current generation too. Here's another of these steady hand applications where Well, we've done a lot of them. Here was a thing I did 12 years ago for using this cooperative control to help evacuate a hematoma. But one of the things you often want to do is create virtual fixtures. Here, this should be a video. I'm surprised why isn't that playing? Don't have the video, but basically what we're doing here, as an example, is we are machining bone in the skull base to get access, for instance, to skull base structures for, say, dealing with acoustic neuromas. Now, the problem is this is very, very tedious because you're trying to machine often through some of the hardest part of the body. By the way, if you machine too far, you could hit the carotid or some other structure and very quickly have a dead patient. So the idea is if you have the robot together with some safety barriers that the robot is enforcing, you can machine down, get access, and let the robot both get rid of your hand trimmer, support the tool, still rely on surgeon judgment, but the robot is enforcing these safety barriers. So this is another thing that we've been very interested in doing. Now, in general, I'm not sure I really have time to talk much about virtual fixtures, but if you wear your robotics hat, this whole question of how do you do constrained control of a robot, either for these hand-over-hand robots or autonomous or tele-operated robots are very, very interesting questions to us. And what we've done is a lot of work on these virtual fixtures. Now, if you look at the evaluation loop, again, a lot of the same technologies that we've had, often you want to run your validation even concurrently as real-time feedback. But here are just a couple other examples. If you look at that prostate, one of the dirty secrets of prostate brachytherapy is the seeds don't stay where you thought you left them. So you need to find out where they really went. If one is in a really bad place, you're kind of in trouble. But often if one isn't in a place where you need one to be, you can go back and put another seed. And so there are various imaging techniques. This is mainly the work of Amit Jain, where we're using X-rays to locate the seeds in a way that is co-located or co-registered with the ultrasound images that can see the prostate. I mentioned the elastography. One of the uses is if you're doing RF ablation. You stick a probe into a tumor and you deposit energy basically to cook the tumor from the inside out. The standard method is a recipe. You say, okay, I stick it in, I cook for 10 minutes, and now I assume I've got a nice cooked meatball. I go somewhere else and cook some more. And occasionally you might have a little thermometer, meat thermometer up maybe on the surface of the liver, or sometimes in the probe. Cook until it gets to 98 degrees C or something. What you'd really like to do is cook until done. So if you use this elastography, what you can do is you can actually see the tissue change its mechanical property as you cook it. It's like testing meat on the grill. And so here are a couple cases, clinical cases, freehand palpated ultrasound, freehand placing needles for RF ablation. And these are what B-mode images look like. Again, the projector is not so good, but you can see the lesions a lot better in elasticity images. And here are the corresponding CTs, postoperative CTs. One other example, this is something not yet published. We have a paper where we'll find out in a few days if it was accepted, where here the application is injecting cement into a bone that is at risk of fracture from osteoporosis. And the larger project, the goal is to do the planning, navigate an injection device into the right place in the bone. In this case, it's up near the femoral neck. Inject cement to strengthen the bone according to what our finite element analysis is. Look at the bone, at the cement that we've actually injected, and compute our notion of the strength of the bone, and then if need be, do some more. What you have to assess this is a fairly sparse set of x-rays. Four x-rays would be typical that you might take. And so what you want to do, this is ground truth. This is the cement ball that, based on what we got from a CT scan post-operatively. We want an intraoperative estimate of that. The standard way would be you'd look at the x-rays, outline the lesion in each x-ray, intersect them from the silhouettes, and you get something that looks like that. What my student Blake Lucas did is, it's a very, very clever method. He's able to get reconstructions that are much more like that. And so this is something that we're really very happy about. It's a 3D level set segmentation done from a small set of 2D projection images. Let me just say a word about the process loop, and then I'll talk. At some point I'm going to run out of people's patience. I'm already driving you out. I apologize. As I said, these systems tend to create lots of data. And you'd like to be able to process that data to correlate what you did with the outcome, and maybe that'll tell you what to do better next time. You know, airplanes are safe because they have flight data recorders. This is an area of quite a lot of interest to the computer science part of medical robotics. And let me just talk very briefly. One of our pilot projects, again, we had some small donor funding, and now we've got also funding from Philips, is to take previous radiation therapy plans where we know the plan, and they often will take a day, and use that database to help us more efficiently improve the planning of a new patient. And the way this planning works today is, you know, here's a tumor and things that you don't want to kill. This is the spinal cord, and these are the salivary glands. A person called a dosimetrist sits down, he specifies a really hairy optimization problem to a computer. Very large, non-linearly squares problem. The computer makes its best solution for the settings of the machine. You simulate the settings of the machine, you look at the dose that you got, the dosimetrist then says, oh hell, or something stronger, comes up with a new plan, specifies a new problem, and you go around this loop until you get something that the clinician will accept. The problem is you don't know really when to stop. You have no idea of how good a job you can do. And it can take all day till you get a plan that is acceptable. So what we want to do is we want to look at, just from the geometry of these organs, the tumor and it's the Euclidean distance it's something we call the overlap volume histogram. You can kind of think of it as the configuration space of the tumor relative to these other structures. We want to get an index into a database of all the patients we've treated before. Now, for quality control purposes, what you do is you say, okay, from my database, is there a patient who we think should be at least as hard to plan, who had a better result in terms of sparing these other structures? And if they're so, then why don't you use that optimization problem on this patient's geometry? Turns out if you do that in a significant fraction of cases, maybe 20%, you can significantly reduce the dose to critical surrounding structures while preserving the same dose to the tumor. The other thing you can do is if you start with sort of the best similar patient and try to optimize from there, you can often get a plan that's about as good as a skilled dosimetrist will eventually get to, but with much fewer tries going through that loop. So we currently have a controlled retrospective blinded study of this right now on our radiation therapy patients. We are hoping as soon as we have that done, we're going to put this in the clinical workflow, and we're very excited by it. I haven't gotten to talk about Greg Hager's work, where what he's doing is he's analyzing surgical motions and trying to segment surgical motions automatically. And then you can use this for various purposes, such as skill assessment. Like here, this is the traces of a not so good surgeon and an expert surgeon doing suturing. And you can use it for skill assessment. You can also use it to compare different instances of the same task. This is something that I've always been really impressed by. Here what we have are two expert surgeons working side by side doing the same suturing. And what happens is if one video gets ahead of the other one will pause to catch up. So this looks very impressive, and it's keeping track. The thing that blew my mind was this result. Here's a novice and an expert. And so, okay, so the expert, the computer knows it's suturing. The thing that amazes me, okay, now the expert's waiting, that but at some point, this guy's going to start moving again. And what that will tell you is the computer realizes the novice finally got through that stage. Now you can clearly use this, see your golf swing. That's the Takeo isn't here, but see your golf swing. You could use this for training. You can use it for skill assessment. Other things, maybe you can use something like this for is to detect the phase of the procedure so you can adjust the help you're giving to the surgeon, but more automatically. Here, I did talk. And this is another result from Nicola Padoy, who is another of Greg Hager's postdocs, where here what's happening is the robot knows what some of the motions of this suturing test could be, and now the robot is doing that automatically. And so now we're back to manual operation, and now we're doing the next stitch. And again, this is one of the directions we're headed. I did talk myself out of time. I'll get done, I promise, before 5. And here is another set of results. One of the things I want to come back to is this notion of a system vision. We got a lot of technology. We can do imaging, we can do modeling, we can do robots, we can do it all. But the key thing is they have to work together. And I really believe, and further, I believe over time, you really need all of the stuff that goes on in the operating room to interface to the much broader information infrastructure of the hospital in the same way that you want your machine tools to interact with, and lithography systems to interact with the information infrastructure of the factory. So I think, again, coming back to this system view, we have here the robots, the imagers, all of this stuff, but in a way, the robot of the future is really going to be the operating room. And I think, again, this comes back, we need to take a systems view. For us, one of the things that we wanted was the ability to put all of our different research operate interoperably in a very highly modular fashion. And so actually working with Intuitive, we've put all of our robotics and medical robotics software into an open source framework that we call SAW, Surgical Assistant Workstation, that we've used for a lot of the things we talked about earlier. Turns out there's a plug-in for the Da Vinci, so that if you develop something, say, on one of our robots, we can try it out on the Da Vinci and vice versa. So I think, I don't know if you do this kind of thing at CMU, but I think it is a very important element of any large research program, and we are really looking for additional collaborators to use and develop this environment. Here's another environment based on some of that C-arm X-ray stuff that again uses these same components for a surgical navigation application, and that was supposed to have been hidden. So anyhow, there you have it, folks. There's the loop. Generally, I think it's really good in doing even sort of basic research to always know why someone would care what you're going to do as well as what you're going to do. And so this is a common model for everything we do. And this, I promise, is my last slide. I've ended every talk for about 15 years with this slide. And what it comes back to is why I changed careers. I honestly believe that this combination of technology and information systems and humans working together can profoundly change interventional medicine in just the same way that we did with factories. It can do it by providing better capabilities, things that you can't do otherwise. You can make procedures more accurate, you can make them safer, you can produce better outcomes. And that combination is actually, even if the machine is expensive, can be more cost effective because you get it right the first time. And finally, ultimately, it's about the patient. What the most rewarding times I've had haven't been getting an award occasionally or getting a big NIH grant or showing off to the dean or the big one. It's been going around with a clinician, seeing a patient where some piece of our technology has been used to help treat that patient. And that is, I think, we should remember the ultimate payoff in what we're doing. We're in business to change society, to help society. And fortunately, you can have fun, you can get tenure, you can do all of these other things along the way, but it's ultimately about that. And this is hard work. An area like medical robotics, you have to know something about medicine as well as something about robotics, so there's more to learn. It takes longer, and it's really hard. But it is fun and very rewarding, and that's sort of the last I wanted to say, unless you want to know about trout fishing. And I do apologize for rattling on too long. I realize I should have been more selective about, or more quick at some of the earlier things I talked about. I'm happy to bend people's ear, answer questions, whatever. None. Okay. Okay. Yeah.
What have you learned about communicating with clinicians?
Oh, well, it's like sailing. It's all vocabulary. To some extent, that's true. If you learn a little medicine, I mean, it helps if you know that otomy means something about cutting. So yeah, a lot of it is patient. One of the key lessons is you got to listen to what the clinician wants and understand his application. Don't let the clinician tell you the solution, although if he will often try, then try to understand why he thinks that's what he wants. But I think for us, maybe the key reward has been is you have to meet actually frequently with these guys who are very busy, and you have to not make them just impatient. But for instance, for the eye surgery work, it's really cool. We have 2 attending eye surgeons meet with us every Friday afternoon. And they're not shy about telling us what's wrong, and they'll try something, and you know, we'll talk about maybe another proposal we want to do, and try things. So basically just stick at it. One of the things I kind of wish I'd done is had the time to go to a year or two of medical school, which when I was young, I wanted to do robotics, and I thought these pre-meds people were really not people I wanted to spend time with, but that's sort of the key lesson. It just takes a lot of time and very frequent communication. With surgeons, these guys are very hands-on. You can describe something and they just won't understand it. If you show them something, they're very tactile people, and so if you show them something, they'll put it in their hands and maybe get the idea and tell you what's wrong with the thing that you've just put in their hands. Now, I don't know, Cam, did you have the same experiences? And Howie had to leave, but I know he did, so. Okay. Yeah?
What do you see as the future of autonomy in robotic surgery? I mean, do surgeons really look for more autonomous robots, or just robots that can help correct problems with?
Well, look, the first law of robotics, of medical robotics, is the surgeon is in control. Personally, I think this whole autonomy versus teleoperation, I'm not sure what it means. You're not going to have a robot walk into the operating room and say, here's the patient, and start carving. I really see it more as trading off one for another. One thing that has driven me up the wall for about 15 years are people who assert that a hand-over-hand controlled robot for doing something like a bone machining procedure is safer somehow than a robot like Robotic, where the surgeon has verified the alignment and now the robot is doing something with some constrained autonomy to cut a shape. The big danger in that kind of robot is not the robot's going to run amok, it's going to do exactly the right thing in the wrong place, so the real safety issue is really a registration issue. I think that surgeons are very conservative, and anytime you want to give them a new piece of technology, other than a few crazy early adopters, what you've actually got to show is somehow it makes their life easier and convince them that it won't do what they don't want to do. Matt, you may be older. You remember DWEM?
Yeah.
It stands for Do What I Mean, which there was a guy who was sort of our generation of grad student, and that was his thesis, was sort of semi-automatic programming. Do what I mean, it would fix your bug. And here it was more nadwatam, don't do what I don't mean. And that's very important. So that's sort of a meta question. Where I think that you can have, kind of autonomy I see actually getting in is you get to some place and now there's some servo-controlled thing that would take over and do some small motion and then go on. I mean, this automatically pulling the suture, I'm not sure is such an example. If you had maybe a third-hand robot actually trying to do something like some simple retraction and controlling the retraction, that would be probably a more plausible scenario. But again, this is all sort of speculation. It's hard to say. Clearly, FDA, it's a big issue with them, and I think you have to be kind of careful in defining what advantage each piece of autonomy is providing. Anything else? Well, again, maybe I should just close again by thanking you folks for sticking for a long-winded talk. Professors, as we get older, talk longer. And also, again, thank many colleagues. I talked about the work, again, of many, many colleagues, and there were many, many funding agencies, both the U.S. government and in some cases private companies and foundations, who helped support that work. And we do look forward to collaboration with you all in the future.